This is Arnold Caplan, and it's my pleasure to contribute to this webinar and provide information about the new MSC, bigger, better, and more clinically useful. And I'm going to focus on the controlling aspects of MSCs with regard to their function in the innate regenerative potential of tissue. So these are my obligatory disclosures. I started a company called Osiris Therapeutics. I have no relationship with that company or any of the derivatives of that company. I receive royalties through Case Western Reserve, which are not enough to pay for the coffee that all of you are probably drinking at this moment. In 1991, I coined the term mesenchymal stem cells, and I'll discuss why I did that and felt justified. But in 2010, I renamed the MSC to be medicinal signaling cells because they function at sites of injury and produce therapeutic molecules. So in bone marrow, you have a hemopoietic stem cell that gives rise to all your blood cells. Also in marrow, there's another cell which I called the mesenchymal stem cell, and I did so because I was able to get these cells to attach to a petri dish, get them to fantastically expand at a high rate, and through using specific inductive agents, I could get them to form bone or fat or cartilage or other mesenchymal cell types. So in the late 80s, I generated this hypothesis slide and again put the MSC at the top and said that these cells could go down these lineages and make differentiated tissues. Again, the dogma of the day is what we saw in tissue culture is what happens in the body, and therefore I felt justified in calling these cells stem cells. So all of this was from observations in vitro, and indeed we did do some in vivo tissue engineering experiments, which also seem to point to the capacity of MSCs to form tissues. So it turns out, fast forward to today, what we see is that all of these tissues I've listed on this slide, which are covered in these publications, all of these tissues, one could isolate colony-forming units, fibroblasts, MSCs, get them to multiply in culture, and then get them to differentiate in vitro down these pathways. What I missed in all of these publications is the fact that all of these tissues are heavily vascularized, and that actually the MSCs do not originate from stroma, they originate from blood vessels. And these perivascular cells, pericytes, give rise to MSCs when this vessel is inflamed or broken. But again, this, it turns out to be the contractile element. When I took my blood pressure medication this morning, this was the unit that contracts or expands to produce lower blood pressure. So again, these are all pericytes. They come from different tissues. And what you immediately are impressed by is the fact that they have different conformations. But you have to remember that all the tissue on this side of the blood vessel is different than the tissue on the side of the blood vessel. we're very clever in getting technologies to release these parasites, and we put them into culture, and we can get all of these differentiated products. Again, this is all in vitro, so MSCs are multi-potential in vitro, but this is not what happens in vivo. And this is a publication which quite proves that. Again, the labeled MSCs can be put into culture and form these mesenchymal tissues. But if this labeled MSC is in an animal which you put on a high-fat diet, so you're making lots of fat, or you break a bone, or the animal ages, none of these perivascular marked cells are found in that differentiated tissue. So therefore, these cells do not function as progenitors for differentiated tissues. And I'll come back to that, a very important point, in a few minutes. So you break a blood vessel or inflame it, and the parasite comes off, and that parasite specifically differentiates into an MSC, and the MSC senses the surround and becomes activated. This activated MSC makes a curtain of molecules that can control the local immune system. So this cell is immunoevasive, meaning it doesn't immediately attract the attention of your immune system. And from the back of the cell, it produces molecules that I call trophic, which are anti-scarring. They control pain, which I'll get to in a few minutes. And they make a variety of medicinal molecules. And they set up a regenerative microenvironment. So we know a lot about what anchors this parasite to the basement membrane of the blood vessels, and the anchoring molecule is PDGFBB. And when you use PRP in various preparations, you're actually using PDGFBB to dislodge the parasite from local blood vessels, giving you an MSC. Again, MSCs in vitro can make these secretory osteoblasts, but more importantly, they make trophic and immunomodulatory molecules, which are medicinal. So again, parasites can be dislodged and taken off blood vessels. They can be activated, and they can make a range of molecules that are trophic, immunomodulatory, or in some cases antibacterial. But more importantly, they set up a regenerative microenvironment. So MSCs have several mechanisms for communication. One is they touch other cells, and so cell-cell communication. They send out vesicles, but more importantly, they send out a range of important effector molecules. So the MSC is hugely secretory. So if you go to clinicaltrials.gov, what you see is that there are over 1,250 clinical trials listed worldwide. This is a map of the United States. I'm very politically active. I don't like red states, but on this map, I love the red states because they have the highest number of MSC clinical trials. This is Minnesota. Can't understand why this cold and desolate state has so many clinical trials. Oh, it's where Mayo Clinic is, and Mayo Clinic's been working with MSCs for over 10 years and has a therapeutic institute for cell therapy. Here's Europe. You can see clearly that Spain is the winner here in terms of the number of MSC clinical trials. They're current. So again, a large array of clinical trials. So MSC, if you add them exogenously at a site of inflammation or broken blood vessels, The MSCs will dock at blood vessels because that's where they naturally do business. There'll be an immunomodulatory and regenerative component. And what they do is they assist the body and the local tissues to set up the innate regenerative sequence of events for those tissues. So MSCs are not stem cells. They're not stem cells. They're medicinal signaling cells. So I continue to use the MSC shorthand way of discussing them, but again, they're not stem cells. And I want to make one other very important point, which is that MSCs are parasites, so they're on all of these blood vessels and all of these tissues. But each one of those tissues has its own unique tissue-specific progenitor cell, sometimes called a tissue-specific stem cell. The hemopoietic stem cell is a progenitor cell. It is not a pluripotent cell. It can only make blood vessels. Muscle stem cells can only make muscle. Cardiac stem cells can only make heart. But the niche where these stem cells are associated is with a parasite on a blood vessel, and they're both in communication with the blood flow. So this is the universal progenitor cell niche. And again, the parasite, this unique cell, is specific to this progenitor cell. I'll come back to that in a few minutes. So what's missed by lots of people is that MSCs are sensory. They actually analyze the microenvironment in which they're situated. Here's a perfect paper for explaining that. This MSC is exposed to inflammatory signals, so it makes anti-inflammatory molecules. This MSC is exposed to bacterial signals. You don't want anti-inflammatory, you actually want inflammatory molecules so you can bring in other cells from the hemopoietic system, macrophage monocytes, to clean out the bacteria. So again, same cell, two different signals, two different, completely different rollouts. So again, MSCs dock at sites of injury, and they assist the normal tissue in regenerating itself. So I'm going to switch gears a little bit because this is a perfect example to explain to you the science of MSCs and why MSCs are going to be important in curing COVID. So this is a science article from March 27, 2020, and it has a picture of the spike protein on the COVID virus, and this spike protein docks in a receptor on cell surfaces called the ACEs2 receptor. So this could be on blood vessel cells, on endothelial cells, or on lung cells, for example. So when this virus, the SARS-CoV-2 virus, docks on the ACEs receptor in a blood vessel, it actually infects that cell, and eventually it causes huge breaks in blood vessels. And this is why we get huge clot formation. This is the same story on lung tissue. It docks on the ACEs2 receptor and punches holes in lungs, and that's how the virus particles can get out and actually infect heart and other tissues on the outside. So what's interesting is that on January 23rd, 2020, over 1 year ago, in Beijing's UN hospital, there was a clinical trial for severe COVID patients where 7 patients were given infusions of culture expanded marrow MSCs. And what you saw is the decrease in CD4, CD8 cells and natural killer cells after a few days, And what's more important is that you saw that the patient was starting to get better. The patient was starting to get better. And in talking to the physicians, after 2 to 4 days, the patients stopped moaning. Pain control. Here are in April of last year, exactly 1 year ago, all of these clinical trials were in play, and some of them have actually reported a rather interesting, spectacular results. Today, there are 110 clinical trials using MSCs for COVID infections, and I want to make the strong point. MSCs can come from dental pulp tissue, from human umbilical cord tissue, from menstrual blood flow, broken blood vessels, therefore MSCs come off, or from, there's a clinical trial for exosomes or for marrow cells. So here's a publication. The senior author is Joshua Hare from the University of Miami, where they use marrow derived MSCs expanded in culture, and they use it on the most critically severe COVID patients. So 9 patients were infused with culture expanded MSCs. 2 of these severe intubated patients died. 7 of them in assays for the spike protein, the MSC infusions knocked down the number of virus particles in the nasal tract of these patients, where in control patients, 31 control patients, they hardly cleared these virus particles. So again, the interpretation was that MSCs could actually, 7 patients walked out of the hospital, could have a profound effect on COVID infections. They're not only immunomodulatory, but they couldn't understand how MSCs could clear virus particles out of the respiratory tract and nasal infections. So MSCs, with regard to COVID, they can mute the cytokine storm. They can repair these holes in endothelial cells or in lung tissue. If bacterial infections start, they actually make proteins that I'll talk about in a minute that kill bacteria. They make molecules that sit on opioid receptors, and therefore the pain goes down and the patients can report decrease in pain. And there's studies now going on at Case Western Reserve that I'm collaborating with, where we now understand that these molecules made by MSCs bind to spike protein or to the ACE2 receptor. These molecules are called defensins. One of them is LL37, and this molecule is indeed antiviral. This is Tracy Bonfield, and with Tracy Bonfield and Aaron Weinberg, who I'll discuss in a minute, we have a publication that discusses the antibacterial effects of MSCs, certainly on cystic fibrosis animals and cystic fibrosis patients. But Aaron Weinberg, who's a dentist, has spent his career studying these defensins, and the defensins are called antimicrobial peptides, AMPs. And Weinberg and his collaborators have characterized a variety of these. I'm going to talk about 2 of these AMPs made by MSCs, LL37 and HBD2, because they bind to the spike protein, to the receptor binding domain of the spike protein. So the receptor binding domain is this green amino acid sequence, and LL37 is this blue amino acid sequence, and it's a perfect fit. So we know now from these computer simulations that there are binding domains on LL37, and actually HBD2 is even better, which bind not only to the spike protein, but they bind the ACEs protein. So that's how they can take and decrease the binding, decrease the number of virus particles in the respiratory tract, because they bind to the spike protein, and also they bind to the ACEs2 receptor. So even if you don't bind to the virus itself, it occludes the binding domain on the cell surface, and also the MSCs produce molecules that. So again, the MSCs function as secretory vessels and also influence the capacity for innate regenerative potential. So I'm going to change the story a little bit. This is one of my favorite tissues, is fat. It has a very rich, as you can see, vascular network, and every single one of these parasites in this tissue actually can give rise to an MSC. So it's clear that adipocytes themselves are secretory. So MSCs and adipocytes could make a very powerful therapeutic cell-based therapy. And what I want to point out is that this paper in 2019 took all of the MSCs from a prep and did single-cell RNA-seq. And what they saw, there were 10 different populations of cells. So the first thing to understand is that all of these MSC preparations are heterogeneous. And here are the populations of MSCs. What these guys did, which was genius, is they made probes for in situ hybridization from these cells. And these probes, so this could be one probe, this could be another probe. These are very tissue-specific probes. And now they did the in situ hybridization, and so the probe for this molecule bound to these MSCs, and the probe for this molecule bound to these MSCs. So these cells represent a different population than these cells. And so now we can tell you exactly where specific MSCs come from. So there's a variety of companies who do MSC therapies. I'm going to talk about lipogens and you have to understand I'm a consultant for lipogens, and I'm a very good friend of Carlo Tremolata, who you'll hear from in a few minutes. So the Carlo designed just an absolutely brilliant apparatus for taking liposuction material, pushing it into the cylinder that contains saline solution. He has wires here to further disperse this, and then he has stainless steel balls in this apparatus, and he shakes these up to further fragment this, and then they turn the stopcock, put saline in to further wash this preparation. The new saline comes in, and all of the monocytes, all of the broken cells, all of the oil comes out into this garbage bag, and they push up on this syringe and collect a population of adipocytes. And the other smart thing is the exit port has 500 micron holes in it, and what you get is a 500 micron aggregate of micro fragmented fat. And actually, this fat has MSCs associated with it and some intact blood vessels associated with it, because 5 days later, when I put these into culture, I can see MSCs at the bottom of this Petri dish. So this cleaned microfragmented fat has both adipocytes and endothelial cells and MSCs in it. And here's a fabulous paper from one of our colleagues, Dr. Promorek in Serbia. And he and his colleagues have 24-month follow-up of OA patients given lipogens in their knees, and they have the 2-year results, which show that the VAS scores are fabulous at this 2-year mark, and you can see the dramatic effects from controls versus those treated with lipogens. So again, my take-home message is that mesenchymal cells in general are secretory, both adipocytes and MSCs. They're not stem cells. These are not, MSCs are not stem cells. Please stop calling them stem cells. They're drugstores for some types of injury. They come from perivascular locations. They sense the microenvironment. They modulate the immune system. They secrete pro or anti-inflammatory molecules depending on the microenvironment. They're immunoevasive, so we can use allogeneic MSCs. They make molecules that sit on opioid receptors and manage pain. They make mitogens for those in tissue-specific and intrinsic progenitor cells, the cardiac stem cells, the muscle stem cells, the liver stem cells, etc. They can help bring in new blood vessels, and they make antibacterial or antiviral proteins, and they have huge secretory activities, and the mechanisms for the long-term action is now under study. What's crystal clear is that MSCs are not stem cells. So my forecast is that 10 years from now, cell-based therapies, MSCs, and others are going to change the way medicine is practiced. These biologics are going to affect the way orthopedic practice is conducted, and they will have medicinal effect on joints and on tissue injuries, and can be used in a variety of both surgical and non-surgical conditions. I teach a course on May 18th to 20th. Unfortunately, May 18 and 19, it's going to be virtual this year. If you write me, I'll give you a free pass to this course. I invite a group of very talented professionals to come and give lectures on cell-based therapy and tissue engineering. Fabulous group. Go to this website and you can see the program. And again, write me and I'll send you a free coupon. This is my favorite Churchill quote. We're at the end of the beginning of cell-based therapy. And from now on, you're going to see more and more cell-based therapy approved by regulatory agencies and brought into practice. All of the work in my lab supported by the federal government, NIH, and it's clear there's a large group of people. These are all the freshmen who line up to let us know what university they're in. These people are the actual workers providing me with data. It's clear I'm only the mouthpiece, and I look forward to the question and answer period at the end of this webinar and to seeing you in person instead of